So welcome everybody. This is Peter Bauer from Centogene. I'm human geneticist and chief scientific officer of Centogene, and I would like to invite you today to join me the webinar dealing with clinical whole genome sequencing. I hope I have prepared very comprehensive and useful information on how we developed this important tool to diagnose patients with rare diseases, and would like to introduce beyond the capabilities we have established to bring you the best knowledge in understanding rare diseases and partnering with us. So just want to briefly start with a short overview on the company. Centogene is a German-based company that is offering genetic services, testing of patients with rare diseases, but as well doing a lot of research on biomarkers, monitoring patients with rare diseases, and ultimately bring as well new therapies and treatment to the patients. Today, on our agenda, we would discuss the opportunities and challenges of clinical whole genome sequencing. I would give you an insight into the value of curated digital data that helps us to understand variants we've seen in genome sequencing, and of course the value of staying up to date and doing repeated reclassification of variants and reanalysis of cases. And in the end, of course, discuss the utility of clinical whole genome sequencing with you. Now, if we look into the challenge we face as human geneticists or a diagnostic service company, every patient has a very huge information set in his genome, which is more than 3.2 billion bases. So even healthy people have differences at more than 4 million of these positions. So the variation in the human population is huge, showing as well the diversity. And for patients, in the end, we have to identify one or very few mutant bases per genome that explain the phenotype and give us an account on the etiology of diseases. You can imagine that such a filtering process from millions down to one or two observations is challenging, and of course needs very tight controls and good algorithms and highly specialized experts to come up with the right conclusion in a decent time. Historically, we used to do direct single gene approaches, which were very good for a couple of defined phenotypes like cystic fibrosis to sequence the CFTR gene, or in Huntington's disease to analyze the length of the triplet repeat expansion. In those cases, already with a pretty high diagnostic yield, but for more complex diseases like, for example, intellectual disability, we almost never did come back with a genetic diagnosis in children with such disorders, and had to wait for better technology. The technology is now available with whole genome sequencing, where we can diagnose almost or more than 50% of the cases, which is a great improvement from almost nothing to more than 50%. However, when applying genome sequencing, of course, we produce those 4 million variants and 200,000 copy number variants. And so there's a risk that we drown in false positive calls that are taken as the cause of the disease, but in fact are not. And of course, still sometimes though we have a very good technology, we miss out the true variant and have false negatives. So the larger the screening project, the more ambitions we have to put into the data analysis to be accurate there. That's an overview. I can share with you how the indication for genetic testing and the right test can be chosen according to a clinical decision tree. And in principle, we have two aspects. The more distinct the clinical features are and the more indicative of family history, the higher is the probability that a single gene or a small gene panel approach will be positive and successful, which offers, of course, a very attractive analysis price, and as well usually is a pretty fast and straightforward selection for the diagnostic procedure. However, the more ambiguous the phenotype or the more diverse the clinical symptoms are, a selection of single genes is no longer appropriate, and then genome-wide screening technologies like copy number array, large panel or exome sequencing or genome sequencing are chosen to find those very rare and dispersed variants that might account for the clinical condition. Now, there's a very important paper of last year, which was put together by the Stephen Kingsmore group in San Diego, and Clark et al. Have done an exhaustive review of published data on copy number analysis on the bottom of the lists, whole exome sequencing studies, and whole genome sequencing studies. And kind of a philosophical term, the diagnostic yield is shown there. Diagnostic yield means the fraction of diagnosed patients among the patients that have been analyzed. And you can, first of all, clearly see that on average, the diagnostic yield in copy number analysis with array technologies is lower than the diagnostic yield in whole exome sequencing, and that one is lower than the diagnostic yield in whole genome sequencing. What you can see on top is that the variation of these studies is huge, and we have, for example, in whole exome sequencing, diagnostic yields between 25%, with the lowest in 119 patients from a Chinese publication, up to 68% that has been seen in a very small cohort with European descent in the literature. Of course, looking at such variabilities tells you something about the validity of such a construct like a diagnostic yield, and therefore I want to make you aware that, of course, for genome sequencing, you have to go for test-naive patients in such studies, because any previous genetic test, of course, takes something out of the potential positive cases and reduces the diagnostic yield. Moreover, of course, different populations will have different a priori fractions of positive patients and influence the diagnostic yield, as does the setup of whether you analyze early onset diseases or late onset diseases. And of course, sometimes even the motivation for exome sequencing in a couple of reimbursement systems forces decisions in the system that have a direct influence on diagnostic yield. For the diagnostic laboratories, and we one among of them, here shown in the Troiano et al. Study of 2017, diagnostic yield of exome is roughly between 30 and 40%. So usually this parameter is taken as a quality of the whole procedure, but I think with the discussion of this data, you've learned that you have caution on that, and it's always important to know about the clinical question, because sometimes we are just asked to exclude a diagnostic a molecular diagnosis, and of course then a yield of 0 is exactly meeting the expectations and cannot be accounted as a bad diagnostic result. However, let's look into the technology and differences from exome to genome sequencing, so that you understand why genome sequencing has higher sensitivity and in the end diagnostic yield, and in the second step, how you can use that for your diagnostic workups. So what you can see here is a screenshot of the BRCA1 gene, where on the bottom we have whole exome sequencing data in one exon 8 of the gene. And you can clearly see that the coding region represented by this square bar is pretty much covered, but not very even. So we have kind of like a mountain histogram of coverage, while on top the whole genome sequencing data set is very even, extends into the non-coding intronic regions in intron 7 and intron 8, and gives us a couple of more variants in the gene that might sometimes be relevant to diagnose a patient. Moreover, you can see in this representation of another patient with a whole genome sequencing data set that a heterozygous deletion, in this case roughly 500 bases, can clearly be deducted from the data set because you see a drop in coverage for that region by 50%. And you can see the so-called split reads that are originating from the deleted allele with this black bar in between. So there's a clear indication of a genetic variant that is sometimes or often not captured by whole exome sequencing because the enrichment process has a strong bias towards losing such sequence information. Another last technical example is this trio genome sequencing, where you can see a very large portion of the genetic information in the region of the GALC gene, which is responsible for Graves' disease. And you can see that the index on top has a drop in coverage, again by 50% on the left part of his sequences, while both parents, father and mother, on the lower panes don't show this drop in coverage, which informs us about a de novo heterozygous deletion, a large one that is often missed by whole exome sequencing and directly represented in whole genome sequencing data sets. Now, I would like to share kind of an interim analysis of Centogene executed on 640 clinical whole genome sequencing cases. These cases have been triggered by different diagnostic scenarios. So in 2/3, we are aware that already exome sequencing and copy number array, sometimes both, sometimes one of each, has been done on that samples, and the clinician asked for the most sensitive test because he clearly expected a genetic diagnosis, while in 37 cases there was urgent need for a diagnosis and the clinician didn't want to miss out something and lose time, and so he primarily asked for clinical whole genome sequencing. There have been solo cases and trio cases, and the origin was sometimes with consanguinity in the parents and apparently sporadic patients with no related parents, and age distribution was as well from newborns to more than 60-year-old patients. So just to show you on the next slide what we learned from analysis of these cohort of samples was, first of all, 2 clinical cases where we came back with a diagnosis in cases where whole exome sequencing might have or actually missed the diagnosis. This is the first one, a young child, 3.5 years old with congenital onset intellectual disability. Whole exome sequencing showed us, again, split reads in intron 1 of ctNND2, representing a deletion of roughly 1,000 bases in exon 1 of this gene. This gene has a high intolerance for haploinsufficiency and is highly expressed in brain, already associated with intellectual disability. In this case, we assumed that the deletion would probably affect regulatory elements that now cause haploinsufficiency of ctNND2 in the patient. To be proven by RNA analysis, so this is only a VUS, but a very suggestive one taken together with the clinical phenotype. Another example is a younger male with 2 years old, with signs of hepatic failure, facial dysmorphism, and a negative whole exome sequencing done before the clinician switched to whole genome sequencing. In this case, we have seen good coverage in the DCDC2 gene. However, the reads abruptly ended in the 5' part of the exon, leaving out a deletion of 75 bases of coding information, and thereby constituting the likely pathogenic mutation in that case. This gene is associated with neonatal sclerosing cholangitis, which matches the phenotype, and essentially these partial exonic deletions are often missed in whole exome sequencing. Now, if we look into the whole cohort more general, we've seen that the total fraction of positive cases after whole genome sequencing for all 640 cases was 31%, which could be disappointing, but in fact is not, because when we stratified for the test naive cases, so the 1/3 the cases where no prior test has been done, we had 41% positive cases, which is in line with the literature and is above the positive fraction of whole exome sequencing. So clearly, there is a 20% diagnostic yield advantage in whole genome sequencing, which of course drops if you have done prior analysis, like you can see on the right side, where those cases that have already been exhaustively studied have only a diagnostic positive fraction of 20%. Nonetheless, you can turn this as well into a message where you can say, even if whole exome sequencing was negative, you will have 20% of the cases positive adding whole genome sequencing, because sometimes coverage is better, sometimes interpretation is better, and both together account for this fraction of positive cases. We didn't see that solo or trio cases have a significantly different diagnostic yield. However, the ambiguity for variants of uncertain significance in solo cases is reduced in trio cases towards a higher fraction of positively or clear positively diagnosed cases. So that's not a surprise, but a reaffirmment of our notion that the more information we can draw into a genetic analysis, and parents are very important in deceased children, the higher is our probability to come back with a definitive diagnosis. Let us quickly think and talk about the number of variants which is used and how we can avoid that this huge number of variants is seen a problem in the diagnostic process for those that ordered the test. So, of course, we need a very high quality data production to have no noise in our raw data set, and that's guaranteed at Centogene by applying the best quality genome sequencing technology available in the market right now, which is the HiSeq X. And of course, we reduce the risk of speculating or interpreting technical artifacts by orthogonal validation. I don't go into details there, but I would like to refer you to this Genetics in Medicine paper we published last year, where you can see how we apply a strict validation of variants for those that do not fulfill quality criteria, and by that keep up sensitivity and specificity of over 99% for the whole process of variant identification. It's as well, of course, important to apply the ACMG guidelines in classification and do some gene curation. And just to mention here, I won't repeat how classification is applied. That's something you can read in the paper. But what you get delivered by a classified variant is a level of trust that the classification of a variant holds strong for the future. And the more defined, like a pathogenic variant, the smaller the error probability has to be. In the sake of time, I couldn't include that, but we have as well shown that, in fact, the classification of our variants in our mutation database has exactly higher quality in reclassification than the one set forth by the guidelines. And thereby, of course, I think we really deliver on the promise that you can trust how we classify and interpret the variants from genome sequencing, but as well, of course, from exome and panel sequencing. On top, of course, it's important that also genome sequencing produces a whole genome data set. We can interpret only genes where we have a genotype-phenotype correlation, which today applies for roughly 5,000 to 7,000 genes. So we will not deliver speculations on any research, and the more if we are asked to do predictive testing, we only include strong evidence or definitive evidence genes in such an analysis, which is a diagnostic prerequisition. That system is put forth by the ClinGen group at NIH, and we greatly appreciate their groundwork on the qualification of genes for diagnostic purposes. I already mentioned that Centogene, since doing diagnostic testing for more than 10 years now, started very early collecting these cases in a mutation database called CentoMD, meanwhile including more than 350,000 cases that have been diagnosed or associated with diseases with more than 7 million variants, and, which is special and important, a global representation of cases from more than 115 countries. So you can expect that even if you send samples from Africa or from South America, we have a repository of information that helps us to analyze and interpret those cases more accurate than others, because be aware, more than almost 58% of the variants are unique in that database. You won't find it in other repositories. Now, this is important, and it's as well important to kind of compare content and quality of CentoMD with public resources like ClinVar or other commercial resources like HGMD. And you can see in this overview that, of course, the overlap of clinically relevant variants is rather small as compared to the total numbers. However, of these shared variants between CentoMD and ClinVar, there is still 12% discordantly classified. The same is true for variants between CentoMD and HGMD. And I will specifically highlight why we think that those variants where we have more metabolic evidence from added analysis in our diagnostic workflow have a higher robustness and quality in classification, and therefore give us an edge on databases where just the genetic evidence is collected. What is extremely important is that the clinical information and the classification of variants can change over time. And the patient has not been diagnosed in the past because just the variant could not correlate it with a phenotype, because the patient did not show the phenotype, might show specifically those symptoms later in life, and then a variant or might be reclassified from uncertain to likely pathogenic. This is at Centogene a consistent and reliable process. So we have established a reclassification program, which gives us, as an ongoing process, a feedback on cases that have been changed. How do you profit from that as a customer? you will always get an update on reclassified variants in the cases we have tested together. And so if this changes the diagnosis, you even get a reclassification report so that you can act on that together with the patient and the family to change clinical consequences from genetic testing. So that's a promise for lifetime, since we think we cannot stop being responsible for a diagnosis we've made in the past. The last part I would like to share with you how we do the validation and functional assessment of variants by just adding metabolomics to a genetic test. And the essential prerequisite there is that we receive patient material not with liquid samples, but with our Cento card, which is in essence a dried blood spot filter card. You can see it on the right side that you only have to drop 10 drops of blood on this device, let it dry for 30 minutes, and then you can send a filter card to our laboratory in Rostock by standard mail. The advantage for us is that we can apply not only genetic testing, so we can do exome and genome sequencing by punching out a small cylinder of this filter card. Now we can also add systematic metabolomic profiles in patients or do biomarker measurements in punches of this filter card and come back with a biochemical validation of a variant. The combination of a genotype and a biochemical phenotype is very, very strong because it's kind of independent of the clinical symptoms. Sometimes it's pre-symptomatic, and in all cases, it gives a very high level of evidence, especially when those markers have been clinically validated. The technology we use is highly sophisticated and has an ultra high resolution, which means by 3D characterization of molecules, we get more than 50,000 to up to 100,000 small molecules identified in a single punch of such a filter card. And of course, now we can correlate these metabolites, small proteins, and other molecules with the phenotype of the patient. How this can work, I will show you in the next slide. But before we go into details, of course, you need to understand how we do the analysis. In this case, of course, we need controls where we excluded a genotype. That's easy for us because we have sequenced all these controls exhaustively and know, for example, that they have no variant in a specific rare disease gene. And then we compare the profiles of the patients or the patient with controls, apply pattern recognition and biomarker candidate research software by integrating this in a large database, and in the end come up with candidates, molecules that are specific for a patient and his disease and not seen in a large set of controls that are negative for variants in the respective disease gene. This is an example here where we have applied the hypothesis-free metabolomic profiling in a patient with an ultra-rare disease. And I will just walk you through this information. The patient presented with leukodystrophy. He presented with epilepsy and intellectual disability, was born to consanguineous parents, and after whole exome sequencing, we've seen that an ultra-rare missense variant in ASA3, which is an acid ceramidase, was found. This variant was never described before and formally was classified as a variant of uncertain clinical significance. However, there was one description published for ASA3 deficiency of a patient with a truncating variant that matched the clinical phenotype, and Centogene on top had analyzed the patient variant in ASA3. So we looked up the biomaterials and did this hybrid mass spectrometry approach for metabolites. And all the identified candidates were analyzed applying a principal component analysis with, of course, then clustering of two patients, the one with the clear truncating mutation and our current case with the missense variant, the parents in yellow, and control samples without variation in ASA3 in green. And you can see that the principal component analysis on the left side perfectly discriminated between the patients, the carriers, and the wild-type controls. Moreover, in a heat map of discriminating candidates, you can see that, of course, a couple of upregulated molecules in patients and a couple of downregulated molecules in patients as compared to controls have been identified. Now, knowing for this gene the functional pathway, which is cleaving ceramide from a high molecular molecule ceramides, identify exactly those molecules piling up in the upregulated as a metabolic block due to the enzyme deficiency. And we could show depletion of a couple of other ceramide molecules, the fragmented small ones, in the depleted part of this heat map. Interestingly, of course, we had many more candidates that were correlated the same way without being discussed or identified in the pathway yet. So this is as well a heat map where we can work on pathophysiology and better understand what happens in that patient on top of the known biochemical deficits. And you will understand that we can apply this technology as well to genes and gene functions that are not yet described, because the clustering and the principal component analysis can always be done and helps us then in the second step not only to do maybe a more rational classification of a missense variant, but as well have a first entry into understanding pathophysiology by applying such a test. Now, to summarize what we have been walking through, clinical whole genome sequencing is often used as the last genetic test offered, and this means, of course, that there is already a small probability of a positive test, but a high impact if such a diagnosis is made, and therefore even the 20% positive fraction in cases that had already prior test might be very important in those cases where genome sequencing has been chosen. I think we should not focus too much on clinical utility because the definition is difficult, but rather talk of or diagnostic yield. We should rather talk of clinical utility because this also includes negative tests where exclusion of a diagnosis was anticipated and achieved. And of course, you've seen that with growing knowledge, better databases, updates of information of classification, which is done at Centogene consistently in CentoMD, you will increase the clinical utility and as well the diagnostic yield over time. Finally, you've seen that while the genetic information is very stable, of course, we have a very dynamic interpretation knowledge, which can be much informed about through other omic technologies. I've shown you the metabolomic value, and this, of course, is also a very important tool to increase the quality of the processes. So to conclude, what you've seen is that with clinical whole genome sequencing, we are already at a very good sensitivity and diagnostic rate, but you can expect that with these data sets and the future knowledge to come, even negative cases of today will turn positive. So the big advantage of clinical genome sequencing, and one of the adverbs in the title is as well that we have a very fast solution because we avoid repeated testing in critically ill patients. We avoid delay in validation of artifacts because the data set has just the highest quality and the least technical artifacts included. And of course, we add more than just single nucleotide variants, as you've seen the examples, especially about copy number variants. The data set is comprehensive and probably stable over years, which we don't see with whole exome sequencing because their technology changes and expands into the non-coding part already. And of course, genome sequencing is a very fast technology because we avoid many pre-analytical manipulations and thereby, of course, offer turnaround times that are unique and as well have entered the Guinness Book of Records, as you probably have read last year. So having said that, I thank you for your interest in this presentation. I would ask you now, first of all, to just use the Q&A function of this webinar to drop me any questions if you have questions. And on top, I want to mention that, first of all, Centogene and our representatives are available out there to answer your questions as well locally. And finally, that we would like to share a questionnaire with you that you can give us a feedback on the content and your satisfaction with this webinar. But as said, please feel free to drop me questions that can be now answered in the next 5 to 10 minutes. And as I can see, there's already one question in which asks, what is the cost of a test if we really produce fast exome sequencing? Of course, there you ask for the most expensive test because when we receive a sample and have process it with urgent speed, because the answer is needed within 72 hours, we have to put this sample immediately on the sequencer. We have relatively high sequencing costs, so this can't be done for less than 7,000 euros. However, the better the technology is developed and the more samples arrive in parallel, better pricing is possible, and that's something we try to drive together with our technology partners in the near future. Another question I received is the question how to balance between exome sequencing and genome sequencing. And we discussed this a lot with our clinical partners, and common feedback is that, of course, you would like to sequence index parent trios whenever samples are available, which is not always the case, but trio sequencing with whole genome sequencing approach is, of course, pretty expensive because you have to add the parents there as well. And so usually then partners tend to go for a trio exome, which has a pretty similar cost price than a solo genome. And interestingly, the diagnostic yield is as well comparable. However, sometimes the richness of information for phasing, of course, when parental samples are added, is an argument for trio sequencing still on an exome basis. Another question I receive is how many genes will be covered in clinical whole genome sequencing. So we interpret genes that are clearly have a genotype-phenotype association, which is for the moment roughly 5,000 to 6,000 genes. Of course, if there is larger chromosomal abnormality, we will report that as well in terms of a contagious gene syndrome, which might be picked up. And for all the other variants, You will always have access for raw data, that's no problem. But of course, a clinical interpretation, if no phenotype is described, is not possible. The question, another question is on autism spectrum disorders, whether whole genome sequencing is useful for this clinical management. Well, I would answer that when you have a diagnostic workflow where you're doing copy number arrays anyway for children with autism, and then case by case decide to do exome sequencing, probably you are very close to doing the clinical genome sequencing as the first one and only test. We have as well a product which is called a panel genomic, where we offer you whole genome sequencing for a subset of genes, in this case, for example, the ID autism genes, to a more attractive price than whole genome sequencing, including the copy number. And in case you don't have a positive finding in this limited but large gene panel, you can open up the whole genome sequencing data set by paying the discrepancy between panel genomic price and whole genome sequencing price. Check, there's more questions. How to interpret rare synonymous variants? That's a good question, because usually if we don't have a good splice prediction signal, we would not assign synonymous variants a score that highlights this for reporting. So usually those variants are classified as WUS, and then they don't get attention into a clinical conclusion. However, of course, the variants are not dropped in the variant list, and we give full account on variants seen in whole genome sequencing upon request. There's another question whether the information about reclassification of data flow back to the clinician who has ordered the test. Patient, yes. So if we have a consent to recontact, and that's specifically mentioned in our consent forms, we will always inform about these reclassified data and variants. And yes, as well, it's the ordering physician that is accountable and will receive this information, usually by email or through our web order page, Cento portal. Is it possible to obtain the raw data from Centogene for genome sequencing in RES? Yes, this is always possible. There's a defined workflow how we offer this option. But be aware that the raw data of genome sequencing is huge data sets, and you need to be prepared to handle such big files in your own IT environment. So I think there's one more. Do you sequence variants in cis-regulome as well? Technically, we sequence these variants. Whether we are able to interpret all these variants, at the moment, I would say information is there, and for some variants, we have already some proof of functional evidence. Then this gets into the report. But of course, many of these variants cannot be systematically diagnostically annotated, and therefore represent part of this, what I called the knowledge for the future. But be aware, of course, the data set is there, and whenever evidence has been given for such variants, we will annotate the variant accordingly, reclassify the variant, and inform you about this change in classification in your patient. So there's a couple of technical questions of how different requests can be done. I would ask you to get in touch directly with our sales representatives or our customer support. And I think for the moment, that's the relevant questions I could see. And there's one question: have you seen any case with multiple genes with mutations leading to almost similar phenotypic features? If so, and how and which gene mutation you prefer? Of course, that's a rare exception from the major findings we have. We had one case, I think, with 3 phenotypes, or let's say 3 clearly pathogenic mutations in 3 genes, and overlapping clinical phenotype reported all these variants back. And of course, sometimes it's very difficult to assign then symptoms to specific genes. So I would not call it a preference. If we have such complex cases, we just have to lay out the complexity. And in such cases as well, we offer direct communication and feedback on those cases just to help you sometimes with complexity that you usually do not expect. And even we sometimes have to digest for quite a while to come up with a good concept how to describe it and how to interpret with the clinical case. I think this was a very active question and answer session. So I thank you for all your contributions, because it helped at the end once again a little bit to describe the framework of what we're doing. As said, Centogene will follow up with a questionnaire, and I highly appreciate if you return those questionnaires to give us guidance on what to improve in the future. Once again, thank you for your interest and your participation today. And I would like to say goodbye and have a good day wherever you joined us. Thank you.